This video is about vegetative reproduction and spore formation in Rhizopus. Here in the live class, we have talked about this very important question. Can all multicellular organisms reproduce like Spirogyra? So we had a discussion on that, which is related to the differentiation level of the organism, the complexity of the organism. It depends on that whether the organism will be able to reproduce like Spirogyra or Planaria. It is very important question. So, but this video is not about this question, so we'll move to the next method of asexual reproduction, that is vegetative reproduction. Example is bryophyllum. The meaning of vegetative here is plant body is divided into two parts, reproductive and vegetative parts. And as it is clear from their name, the reproductive part will help in the reproduction and vegetative part help in the growth and maintenance of the plant. But when the vegetative part like root and stem, leaves, they help in reproduction. Their their primary job is not to do reproduction, but when they do, such reproduction is called vegetative reproduction. An example is leaf of bryophyllum. Here you can see in the notches of leaf, buds are growing, the roots are there, leaves are there, and when they acquire a particular size, they fell up from the leaf, they detach from the parent plant, and they develop as independent plants. Now, what are the advantages of this method? That we can use vegetative reproduction in plants where seeds are not developed properly. We call such plants as seedless plants, for example, banana, oranges. Oranges depends on the variety whether they have seeds or not. Second advantage is in the formation of genetically similar plants. If plants are of very good variety, so we want to maintain that genetic combination. So if we don't want to change the genetic material, we want to keep that same in all the plants, then we can use this method because in the process of seed formation, which is the sexual method of reproduction done by plants, their meiosis take place and gene combination changes. So all plants are not genetically similar if they are produced by sexual reproduction. But here, in case of vegetative reproduction, all plants are genetically similar. So it depends on the requirement whether we want genetically similar plants or dissimilar plants. Next method is reproduction in fungi. It is by the method of spore formation. Example is Rhizopus. Here you can see this lower part is hyphae, it is the plant body, and on this you can see the spore forming part, stalk and round structure is there. This round structure, if one structure is called sporangium, and if we are talking about more than one, then we call it sporangia. So sporangia for plural and sporangium for singular. Their function is to form spores. Here you can see all these sporangia, they are intact, but one is splitting open and releasing the spores. So this one has matured and it is releasing the spores. It is not known whether the spore will get suitable environmental conditions when they are released or not. So they are prepared for unfavorable condition, and a very thick wall is present around the spore so that it can survive if it does not get proper conditions for the growth and further germination. So once it gets air and whatever uh, uh, factors needed by the spore for its germination, then this wall will break open and the spore will form hyphae. This will make hyphae. Each spore will form one hyphae. So it is a method of reproduction, but asexual method of reproduction because here only one parent is there. Next and last method in asexual reproduction is tissue culture. A very simple method we will talk about. We will not go talk about modification and all. So here you can see in this glass plate we have taken plant tissue, and if we refer to the text, They have told us what type of tissue should be taken for plant tissue culture. Yeah, tissue or cells from the growing tip of plant, that is apical meristem. From there this tissue is taken, then it is transferred to a suitable growth medium. The medium is artificial medium, is growth medium, which will have nutrients for the growth of these plant cells. And you can see irregular growth is forming here on the tissue. Cells are dividing and forming irregular growth here. This is called callus, but we have to make plant out of the callus. So we will shift this callus to a next to some another jar which will contain growth medium, artificial medium. The composition of this will be different from this medium. It will have some hormone that will help in formation of roots. If I zoom it in, you can see formation of roots here from the callus. But we need shoots also. So next we will shift this to another medium. Now this medium is different from this medium because it contains hormone for the formation of shoot system. Here you can see shoots are formed from the callus. So in this way plants are formed from a tissue, not from a seed. Seed is sexual mode of reproduction, and this is asexual mode of reproduction. What are the advantages of doing tissue culture? That we can get many plants from one parent in a short time, which is not possible in if we go by the sexual mode of reproduction by seed formation. Then the number of plant formed are limited. It is equal to number of seeds, but here we can form as many calyces as we want. So in very short time we can grow many plants from tissue. Another advantage is that these plants are disease free because we are taking a growing tip, we are taking a meristem which is actively dividing part, and it is not possible for virus to grow in dividing cells. Viruses, they cannot grow in dividing cells, and that's why we are taking this part. We are taking apical meristem so that we can produce disease free plants. So these are the two advantages of tissue culture. So we talked about method and advantages. So this much about the asexual methods of reproduction in plants. Thank you.